So the first pathway by which UV damages the DNA was the formation of cyclobutane pyrimidine ring, pyrimidine dimer. The second pathway, which accounts for most of the remaining UV-induced DNA damage, produces 6,4 photoproducts. You have seen the CPD accounts for about 75% of the total UV damage to DNA. So the remaining 25, that is contributed by this pathway in which 6,4 photoproducts are formed. A bond is formed between the C6 atom of 3' pyrimidine, that may be thymine or cytosine, and the C4 atom of the 5' pyrimidine, which is usually cytosine. So see here the bonds, these are not formed between C5 and C5, C6 and C6 as it is formed in CPD. But here this bond is formed between C4 and C6 of 2 pyrimidines. So here you can see this is normal DNA and 2 thymine are present adjacent to each other. So UV can damage this DNA by two ways. One is the cyclobutane thymine dimer formation, and the secondly through 6,4 photoproduct formation. So in the first case, you know, a dimer is formed and two bonds are formed between C5 and C6 atoms of adjacent pyrimidines. But in case of 6,4 photoproduct formation, the bond is formed between the C6 of this thymine and C4 of this thymine. So this bond is formed between carbon number 6 of one thymine or any pyrimidine and the carbon number 4 of the other pyrimidine. So in this way, by the formation of this bond, a dimer is formed. So this is a dimer. So both these are dimers, but the mode of formation of these dimers is different. So the 6,4 photoproduct causes a major distortion in bDNA because the two pyrimidine rings are perpendicular to each other. So by the formation of this bond between C4 and C6, the two pyrimidine rings become perpendicular to each other. So that's why it causes a major distortion, in contrast to a minor kink which was produced by CPD, which was a kink of about 30 angle rotation. So if not removed, a pyrimidine dimer or a 6,4 photoproduct can interfere with the normal operation of the replication and transcription machinery, and this interference results in mutations and cell death. Even if the lesion is removed, the lesion caused in the DNA, even if it is removed, the result can be a mutation.